Welcome to the General Chemistry 1. This section is organic compounds, focusing on the structures of biomolecules, particularly nucleic acids. In this lesson, we will describe the structure of proteins, nucleic acids, lipids, and carbohydrates and relate them to their function. Nucleotide components. Nucleotides are the building blocks of nucleic acids. Each nucleotide is formed from a 5 carbon sugar, a nitrogenous base, and phosphate ion. Nitrogenous bases in nucleic acids belong to two families of nitrogen-rich cyclic compounds, which is purines and pyrimidines. So the bases in DNA is adenine, guanine, cytosine, and thymine, while the bases in RNA is adenine, guanine, cytosine, and uracil. Nucleoside versus nucleotide. Nucleosides are made up of deoxyribose or ribose, sugars bonded to a nitrogenous base. Names in red are nucleosides and DNA, while those in blue are RNA. So, illustration right, we could see that the red is named deoxyadenosine, R goes to H, or, and in blue, it is named adenosine, R goes to OH. Now, in the left side, the name in red is called deoxycytidine, R equals H, while in blue, it is cytidine, R equals to OH. And the name in red is deoxythymidine, and the name in blue, it is called uridine. Deoxyguanosine, R equals to H, or in blue, it is guanosine, R equals to OH. The structure of nucleotide is like that of the corresponding nucleoside, except that the hydroxyl group at carbon 5 is replaced with a hydrogen phosphate group. For simplicity, the general structure of a nucleoside and a nucleotide can be depicted as follows. So, a nucleoside, it is formed in pentose sugar, nitrogenous base, and hydroxyl group, while a nucleotide, it is formed in pentose sugar, nitrogenous base, and a phosphate group. Phosphodiester bond. One nucleotide is linked to another nucleotide in a DNA or RNA by a phosphodiester bond. The phosphate group links the carbon at the 5' position of one nucleotide to the carbon at the 3' position of the adjacent nucleotide. The double-helix structure. If a polypeptide chain protein has an N negative and a C terminals, a polynucleotide strand and DNAs have a 5' and a 3' ends. During the formation of the double helix in DNA, the two polynucleotide strands interact in an anti-parallel manner. The base near the 5' end pairs with the base near the 3' end. The two polynucleotide strand in the DNA forms a double helix due to strong interaction between bases from each strand. Each base has its corresponding pair, adenine with thymine and guanine with cytosine. Interaction between base pairs is possible by hydrogen bonding. So adenine forms two hydrogen bonds with thymine, while guanine has three hydrogen bonds with cytosine. The double helix structure. With this specific base pairing, the two DNA strands in a double helix are complementary with each other. A different base pairing in RNA. In the case of RNA, cytosine-guanine pairing is observed, but uracil is the base that pairs with adenine, of the DNA strand since RNA does not have thymine. To summarize, nucleic acids are made up of units called nucleotides, with each of which is made up of sugar moiety, a nitrogenous base, and phosphate ion that are linked together by phosphodiester bonds. DNA strands interact in an anti-parallel manner and are complementary with each other. The double helical structure in DNA is stabilized by hydrogen bonding between complementing bases from each polynucleotide strand. So we have here our references and bibliography for further readings. Thank you. This quiz is on structure of nucleic acids. Let's begin. Thank you.